Hi, my name is Georgina and I'm carrying out my PhD research at the Center for Alternatives to Animal Testing. Our research focuses on studying the effect of compounds on the nervous system in vitro. This is, not using animals, but a cellular system. There has been concern that there has been an increase in neuronal disorders, especially amongst children. Therefore, there's a need for better models to study the complexity of toxicant-induced neuronal damage. The increased use of pesticides, supplements, and other compounds make testing a challenging process. In our lab, we have developed a three-dimensional model to understand the molecular changes which occur during neuronal toxicity. The in vitro systems we're developing in our lab are based on human-derived cells. Specifically, we use a commercial cell line called Lumus and induced pluripotent stem cells derived from human skin cells. Both these cell lines are grown on a shaker and differentiated into neurons to create mini-brains. Mini-brains are spherical structures made up of neuronal cells. These spheres are three-dimensional. Unlike conventional two-dimensional in vitro models, these are able to better recapitulate neuronal development, three-dimensional brain structure, and interactions between cells, just as in live animal brains. In the future, these models could replace expensive and timely compound toxicity testing on animals which induce pain and discomfort. These models are also more relevant to human toxicology and could allow for screening of thousands of compounds to test whether they can induce neuronal toxicity and whether they pose a public health concern before they're marketed. In our experiments, we expose the mini-brain models to compounds and assess the effects these may have over neurons. We use the latest technologies to analyze changes in neuron size and growth, gene expression, cellular energy, as well as metabolism changes. We also use high-end microscopy imaging to visualize these changes. At the Center for Alternatives to Animal Testing, we use a novel technique called metabolomics analysis. This method is used to study changes in cellular metabolism, encapsulating all the changes which a compound may have caused Upon sample analysis, we identify which cellular metabolites have been altered by exposure to a compound. With this information, we aim to identify the molecular pathways which lead to such changes. Our model has been shown to be a promising alternative to study the effect of neurotoxic compounds at a cellular and molecular level.